Okay, welcome back. So in this section, we're going to talk about lethal alleles. If something is an essential gene, it's going to be absolutely required for survival. And the absence of this particular protein product will lead to a lethal phenotype or cause that organism to die. And it's estimated that about 1/3 of all of our genes are going to be essential for survival. Non-essential genes are going to be those that are not absolutely required for survival. So these may lead to something that might not have a phenotype that's incredibly desirable, but it is still going to allow that person or that organism to survive. So how do they become lethal? Well, many of these lethal alleles are going to prevent cell division, and so this can kill an organism at a very early age, oftentimes before birth, just very early in development. There are some lethal alleles that can exert their effect later in life. One example of this would be Huntington's disease. And so this is characterized by progressive degeneration of the nervous system, dementia, early death. The age of onset of this disease is usually between ages 30 and 50 or so, with an average onset at about 38. So sometimes scientists may want to study different alleles that could be lethal, and one way that they can do this is by creating what's known as the conditional lethal alleles. And in these, they're only going to kill an organism only when the certain environmental conditions prevail. So one example for this would be a temperature-sensitive lethal, or TS lethal. And if you want to study an organism and see what a gene does in the adult, but this gene is also required for development, if you not just knock it out genetically, you're not going to be able to see what happens in the adult because they're never going to survive to the adult stage. So if you make it so that it will only be deleted or only be, the expression will be reduced only at certain conditions, you can allow it to grow, develop normally, and then change it once it's an adult to see what the effects might be. So for example, developing Drosophila, the larvae may be killed at 30 degrees Celsius, but will survive if you grow it at 22 degrees Celsius. So you can let it grow at 22. Once it's reached its full adult age, then you can increase it to 30 degrees and see what the effects of that allele might be. And once it's adult, it may not be lethal anymore as well. So when you're thinking about lethal alleles, they can affect the Mendelian ratio. If those alleles are lethal, those organisms may not survive, and so the resulting offspring may give you a somewhat strange ratio that may not make sense at first. And so let's go through an example. And the example here is crossing an agouti mouse, and the agouti is just the color that they call it. It's like this brownish grayish color, crossed with a yellow mouse. So let's look at cross A. If you were to cross an agouti mouse with an agouti mouse, on the F1 generation, 100% of them going to be A or agouti. They're all going to 100% survive. Okay. Now let's skip to just the agouti mouse times the yellow mouse. Okay, so this is a dominant times the yellow, and what you can see here is in the F1 generation, 50% of them are going to be the A agouti, and 50% of them are going to be yellow with one A and then the one A with the yellow. 100% of them are going to survive. Half are going to be agouti colored, half are going to be yellow. Now, if you look and you were to cross a yellow mouse with a yellow mouse, and in this case they both have one A allele and then one with this little y superscript, what you would see is you would expect a typical Mendelian ratio. 25% or 1 in 4 to be agouti, 50% here, or 2 in 4 to have this heterozygote or intermediate. In this case it's a yellow, one A and one of the yellows. However, in this case, the 25% that you would expect from this cross, having the A with the Y and with the Y here, this happens to be lethal. And so you're never going to see these mice being born. And in this case, the ratio of the offspring that you will see is 2/3 yellow to 1/3 agouti. And what you're looking at here is just the survivors. Okay. There are some other cases where you can also alter the Mendelian ratio that you might expect. And you can get this when you combine maybe two different gene pairs that happen to have two different modes of inheritance. So for example, we can look at pigmentation. This is going to be inherited through just simple Mendelian fashion, dominant, recessive, and so on. And you can also include blood type, which we just talked about before as having a codominance effect. And so that's going to be a little bit different than the typical Mendelian genetics. So if you were to cross an individual, I'm just looking at pigmentation here, if you were to cross a heterozygote with a heterozygote, what you would expect is 3/4 of the offspring to be pigmented, and this would be a A or the heterozygote, and you would expect 1/4 of them to be albino or a homozygous recessive. Now, if you were to look at the blood type, and you were to cross an individual who is AB, type AB, with an individual who's type AB, what you would expect to see is that 1/4 of them would be blood type A, 2 out of 4 of them would be blood type AB, and 1 out of 4 of them would be blood type B. And you can set up the Punnett square to see that for yourself. And the way that you can do this is you can use the forked line method to calculate the final phenotypic ratio. So the probabilities, if you were to write these out and you multiply them out, 3 out of the 16 would be pigmented type A blood, 6 out of the 16 would be pigmented type AB blood, 3 out of the 16 would be pigmented type B, 1 out of the 16 would be albino type A blood, 2 out of the 16 would be albino type AB, and 1 out of the 16 would be albino type B. And so this would give you a final phenotypic ratio of a 3 to 6 to 3 to 1 to 2 to 1. Okay, so it's not something that you would easily remember, but by calculating it through the forked line diagram method, it should be fairly simple to calculate. So sometimes we have what's known as epistasis, or expression of one gene pair masking or modifying the effect of a second gene pair. And one simple example of this would be looking at the red hair allele as well as dark hair color. So if you have a very dark hair color, it can basically mask the expression of a red hair allele that you might have. If an individual happens to have lighter or blonder hair color, it can shine through a little bit more easily, and you can have red color hair. This is, again, completely separate genes here controlling two different things. If you have maybe brown hair, it's going to shine through a little bit. You might have an individual who has, say, auburn colored hair. And if you have very dark hair color, you might not see any expression of that red hair allele, although it can still be carried. Okay. So an example of an enzymatic pathway, okay, you might have, say, a colorless precursor, okay, requires enzyme C, and then it's going to have this intermediate, then it's going to require enzyme P, and that can make this purple. So if you're thinking about a flower, okay, you might have this start as white, okay, it goes through this intermediate, and then it goes through the second step, and it can turn purple. Now, if an individual can be homozygous for either recessive allele, either this one or this one, doesn't matter really which one, it's never going to make this final purple pigment. And so what can happen in this case is, even though this enzyme P might be completely fine, you're never going to get, if enzyme C is screwed up, you're never going to get this colorless intermediate, so enzyme P can never work on it, you're never going to get the purple pigment. And so this enzyme C is kind of obscuring the expression of, say, the enzyme P. And so what's going to happen is that, in this case, the flowers would remain white. You may also see sometimes novel phenotypes. So in summer squash, if you have two homozygous dominant traits, you would have this disc-shaped sort of squash. If you have the two homozygous recessive, you have a long-shaped squash. This looks to me like a normal squash and grown in the garden. If you were to cross these two, such that you had one dominant and one recessive, either one, doesn't matter, either the dominant A, recessive B, or dominant B, recessive A, you can get this sphere shape. Complementation analysis is often done in research, and the idea behind this is it allows you to determine whether two independently isolated mutations occur within the same gene, or whether they represent mutations in separate genes. So let me give you an example. Say you have two researchers or two labs, and they happen to independently isolate and establish a true breeding strain of some wingless Drosophila. And they both conclude that it is due to a recessive mutation. If you wanted to see whether that mutation, they happen to just get different mutations in the same gene, or whether it was mutations in different genes, both happen to be involved with wings, they can basically cross these flies with one another. If it happened to be two different genes, okay, let's say fly 1 has a wingless gene 1 and a good gene 2, and fly 2 has a good gene 1 and a wingless gene 2, if you were to cross them, they should complement one another such that their offspring should get one good copy, and I use the word good colloquially, but one good copy of each gene, and then their offspring should have wings because of this. Again, assuming that this would be a homozygous recessive previously, and that the dominant would be that they would have wings. If it was the same gene, it really wouldn't matter if you cross them together, because regardless of which version they're both getting bad versions or bad copies of that same gene, and all of their offspring would be wingless. Sometimes expression of a single gene can have multiple effects, and so the term for this we use is Pleiotropy. And what it means is that the expression of a single gene just has multiple phenotypic effects. And so it can affect more than one thing. And this happens quite often sometimes when a gene gets messed up, it's not just messing up one thing, it's messing up many, many different things that it's related into. An example of this in humans would be Marfan syndrome, and there's many others. This happens to be due to a defect in connective tissue. So anywhere that connective tissue is important, you may have issues. And so this can present with things like eye issues, increased risk of aneurysm, lengthened long bones, all kinds of seemingly disconnected symptoms. But they are connected because they're all involving connective tissue.